Stanford University.
Well, good morning. It's such a pleasure to be here and talk to you all this morning. So as you heard in the introduction, I would like to talk to you about rejuvenation and regeneration, a subject that humanity has dreamed about since we have recorded history. This is the legendary Fountain of Youth, a painting from Lucas Cranach the Elder from the 16th century. So this Fountain of Youth works as follows. You're old and frail, or you have a disease. You enter the fountain, maybe you drink some of its water, and you enter young and rejuvenated on the other side. Isn't that great? Wouldn't it be wonderful if we had something like that? Now, we know, of course, that this is an elusive dream, but it's interesting, as I said, every culture, every civilization had ideas of this fountain of youth, eternal life. And for example, Ponce de Leon spent much of his life chasing this, or Alexander the Great. They were chasing this fountain of youth, and actually in Florida you can visit some of these presumed fountain of youth. You can drink the water, but it doesn't have big of an effect. But what I would like to discuss with you today is an absolutely remarkable development in aging research that may change the way we think about aging and how we may treat age-related diseases. And this started with a model called parabiosis. In this model, two animals are surgically connected, specifically a young mouse with an old mouse in this example. And after a few days, their vasculature joins so that the young blood now can enter the organism of the old mouse, and vice versa, the young mouse will be exposed to the old blood. And this is, in some extent, not unlike Siamese twins in humans, although they can be attached at different places, except we induce this surgically, we suture them basically together. And I know that's a bit creepy, it's a bit Frankensteinish. But it's an absolutely amazing model. It was developed actually end of the 19th century in France and was then used to discover, for example, that there are viruses that can induce tumors from one animal to the other. It was instrumental to discover some of the factors that determine sex, female or male, as well as some hormones that are involved in obesity. But a few years ago, Tom Rando, who's a faculty here, also in neurology, was the first to use this model to study stem cells in muscles specifically. So he was wondering if you're old and you have a muscle injury, your muscle actually doesn't heal well. It produces mostly fibrotic tissue that is not useful to regain muscle activity. But what he found, if he exposed an old mouse and injured its muscle to a young mouse, that old mouse could regenerate its muscle almost like a young animal. And he discovered that this was due to some of the young factors that are supplied to the old mouse. This was reproduced actually recently, last year, by a scientist at Harvard, Amy Wagers, and then others found similar effects on the pancreas. Tom also discovered effects on the liver, and others found effects on the heart. So all these organs in these animals, in old animals, can be regenerated and rejuvenated. Not entirely made young again completely, but they clearly look like a younger organ. Now, what we were most excited about is to apply this to the brain, to ask whether an old brain could be regenerated or rejuvenated. And my lab, together with a very talented graduate student, Saul Vileda, who has now his own lab at UCSF, and then a couple of other people since, have been able to show that indeed the old brain can be regenerated by exposure to young blood. And I repeat, the old brain, simply exposed to young blood, looks more like a younger brain. And that was just really amazing. And so what I want to show you is sort of the ideas behind it, how we arrived at this, and how this might work and how we might potentially translate this to humans. So this slide here shows aspects of cognition. On the x-axis here you see age, and then different aspects of human cognition such as reasoning, spatial orientation, perspective. We don't really know why this is. We know that with age, the connections between nerve cells in our brain, the synapses, they start to deteriorate, they start to degenerate. The neurons, the nerve cells, they start to die, they start to shrivel up, and ultimately the brain actually shrinks and it becomes more susceptible to diseases such as Alzheimer's or Parkinson's disease. So what we would like to do is to stop that process or even reverse it and ask the question, can we regenerate or rejuvenate the brain, the old brain, or stop it as it becomes older? So how can we study these processes? One of the biggest challenges in studying the brain is that it's in the skull and it's not easily accessible. Yes, we can do behavioral tests, we can interview people, we can do all kinds of intelligence and memory tests and things like that. We can use imaging, increasingly sophisticated, but most of these cannot look at the molecular events that take place as we get older or as the brain becomes sick and degenerates. So largely the brain is inaccessible to molecular studies. And we have to wait usually until a person dies and volunteers to donate their brain for research. And then we can look how did the genes change, how did the proteins change in that person with the disease. But that's after the fact. So we don't know how it leads up to that. So as we get older, lots of different things change in our body. And aging researchers have identified what they call here in this fascinating review, hallmarks of aging. And you may have heard of some of these. For example, the DNA becomes unstable. You start accumulating mutations. The telomeres, the part of the DNA that is necessary to replicate the DNA becomes shorter. There's changes in the DNA that accumulate with age. We call these epigenetic changes. The proteins don't function as well anymore. They start to accumulate. You may have heard of amyloid that accumulates in the brain. We call this loss of proteostasis. Nutrient sensing becomes dysfunctional, and so forth. What we were most excited about is one aspect of aging that people are well aware of, but it's still not clearly understood what happens, and it has not been studied at a broader scope. And that's called intercellular communication. And I will talk a bit more about this. So what does that mean? It's in a way how the cells in our body communicate with each other, how they talk with each other. I often call this the language of cells. That language seems to become garbled as the organism gets older. And maybe the cells don't quite understand anymore what they're trying to say. And so what we try to do is to test the hypothesis whether changes that occur in the brain with age in the organism, whether they're related to changes in the rest of the body. So because we don't have access to the brain, can we look in the rest of the body? And as the organism gets older, does that affect the brain? So is the organism connected with each other, and the factors that mediate the communication between different organs in the body, do they maybe change, and can we study them? So at the heart of this communication is the blood. The blood is the single tissue that connects all the organs in our body. If you don't have blood supplied to an organ, it will die very quickly. So, of course, we know the blood carries cells, such as red blood cells that carry oxygen. It also has white blood cells that fight infections and so forth. But it contains a lot of proteins, a lot of factors that are involved in this cellular communication. And so, is the blood mediating the communication between muscle and brain, for example, that we think might have a role in exercise, for example? If you exercise and you use your muscle, there seems to be a benefit to the brain. Liver with muscle, pancreas with liver, and so forth. Is the blood conferring this information between tissues? And then, consequently, as the organism gets older, does the blood age as well? So if you look at young blood versus old blood, are there changes that take place that we can potentially measure? And so we have two questions that I want to discuss with you today. Does blood regulate aging? And can we find factors that might mediate these effects? So I will show you a very interesting experiment that we did a number of years ago, and we're still working on that and trying to test this hypothesis that there are factors in blood that can be used to both understand aging and potentially modulate aging or even predict how old a person is. So we got samples, blood samples, from roughly 300 individuals. These were all cognitively normal people from age 20 to about 90 years of age. So they were seen actually in memory centers across the country, and we obtained these blood samples. We use only the soluble fraction, which is also called plasma. So we get rid of the cells and we just get the soluble fraction. And then we use the tool called proteomics, which basically means the art of measuring proteins. So we measured lots and lots of proteins. And again, what kind of proteins are we interested in? We're interested in proteins that cells use to talk to each other. So if you have cell 1, for example, it can release a factor, for example, an interferon or a growth hormone, can release this factor into the extracellular space. So it secretes it outside, and then this factor may reach a neighboring cell, cell 2, or in between here, maybe the blood that takes this factor, transports it, and whichever other cell has what we call a receptor can receive this signal. And once this cell receives the signal, it may do different things, and these are just examples. This factor may tell the cell, everything's fine. Here is a factor that keeps you alive and healthy. It helps you thrive and be functional and do what you're supposed to do. It may tell the cell to make a copy and divide. Or it may tell the cell, differentiate and produce a sophisticated neuron, for example, from a precursor cell. Or it may even instruct the cell to die because we can't use you anymore, you fulfilled your function. So if we look at lots of these factors, scientists have called them cytokines, chemokines, growth factors, neurotrophins, or hormone-like proteins. These are categories, but basically all these proteins do the same thing. They confer information from one cell to another, from one tissue to another, or within a tissue. So if we look at many of these, we can call this the language of cells. And in this specific experiment, we looked at roughly 100 of these words, you can even call them. So they contain very specific information. And so we had these 300 samples. We measure 100 proteins, 100 words of that language, measure their absolute levels in the blood, in the plasma from these individuals. And then what we noticed, so remember we had from 20-year-old up to 90. When we compared the youngest people with the oldest people, we were really surprised to find that roughly a third, or even more than a third, were significantly different between these groups. What that suggests, what that tells us, is that our body lives in a very different environment when it comes to this language, when it comes to these factors. So it has to somehow adapt. There might be more of a factor or less of a factor, and somehow our body has to adapt to that. So there seems to be an aging signature that evolves as you get older. And what we can do using bioinformatics tests is to pick out those factors that best correlate with age, that best predict age. And you can then take the relative levels of these top factors, in this case 9 factors. We can, for each person, we get the relative level of these 9 factors, and then we can calculate, through regression modeling, we can calculate what the relative age of a person is based on the levels of these factors. So this is how this looks. So here, these are all the 300 individuals. Each dot is one individual, and on the x-axis, you have the actual age of the person, the chronological age. How many years did they live? And then we take, for each person, we take the top factors, these proteins, that we know correlate with age, and we calculate back what is the relative age, and we can call this the biological age. Now, there's still a hypothesis, but it's very interesting. If you see, obviously you would expect, because we took the ones that best correlate, that you have a really good correlation overall. But what's really interesting here are the outliers. And so if we look at this individual here, for example, this is a 70-year-old person, but the proteins, these top aging proteins, predict that this person is only about 45 years of age. So is this a person that looks much younger, has maybe a very high life expectancy, will live up to 100 years of age, will be free of age-related diseases, of cancer, and it will just be much fitter than its peers? And then on the other hand, you have individuals like this one, who is not even 40, but seems to have an aging signature of 65 years of age. So is this person at risk to develop an age-related disease? Maybe looks older, maybe doesn't feel as well and as young as it actually is. So this is still part of an ongoing hypothesis that we're testing. We're trying to find better factors that can better predict, but you can easily imagine, if this works, you could actually test people as they get older and sort of manage how old their relative body is, right? What is your relative age? And recruit them into regimens to maybe live healthier or do screening for very specific age-related diseases that such a person might be at risk. And there's a big interest in aging research, as you might heard. There's this new company, Calico, for example, that are for the first time trying to address aging from a pharmaceutical perspective. So to understand aging at the molecular level, and then potentially find people that have accelerated aging and enroll them into clinical treatments or life interventions as they get older. So we're incredibly excited about this possibility and trying to understand this better. And I give you one example here. A few years ago, we had a publication with Saul Vileda, who I mentioned already, that got a lot of attention because we used this parabiosis model and we described some factors that seem to accelerate aging, at least in these mice. And one of these factors is called CCL11 or Eotaxin. And you don't have to remember this name, but it's just an example of a age-related inflammatory protein. And this factor, as an example, it gets higher and higher as people get older. So you have more and more in this blood. And we think it has some detrimental effects. Now, our body doesn't make anything that is inherently bad, but it may get at levels that are too high for what it's supposed to do. Also, if we take spinal fluid from individuals, and again, these are healthy individuals, there seem to be higher levels of this factor. We're really excited about centenarian studies, and this is a collaboration with Claudio Franceschi at the University of Bologna. And he has this amazing cohort of individuals that are, first of all, centenarians or even supercentenarians. So supercentenarians are, I think, past 105 or 110 years of age. So he has this cohort of 80 of these individuals, and then he has their children. Now, not all of them, but you can see 63 of these also have children. And so that's sort of a genetic control, right? You would expect that the children of these centenarians, at least some of them, will also live to a very long life. We know lifespan and health in general is inherited, not completely, but to some extent, let's say 50%. For argument. So we have these kids of the centenarians, and then he has also the spouses, which we would call an environmental control or an epigenetic control. So these don't have the genetic material of the centenarians, but they live in the same household as the offspring. So can we use such samples, for example, to ask, are there these proteins that we find that we think predict aging or are strongly correlated with aging? Are they different in these same age people? So they're both the same age, both cohorts are the same age. But these would be predicted to live a long life, at least some of them, while these would not be necessarily. And so we look again at this factor Eotaxin, and just to remind you, it goes up with age. Now, if we look at this, we look at the spouses, and the average of the spouses is here. It seems the offspring already has an almost significantly lower level of this factor, and clearly the centenarians have yet lower levels. And I just told you, with age, this factor goes up. It's a bad factor. It keeps going up and up. But somehow it seems the centenarians manage to keep it low, and some have it really low. So did they find a way to cope maybe with some of these aging factors, or just genetically they have lower factors, and that's why they live longer? And in the same rationale, we want to also find factors that have beneficial effects. So they have levels of beneficial factors that are higher, and so they live longer. So these are some of the experiments that we're working on now. We have actually many more of these. We have almost 500 samples from this cohort, and we want to measure more and more of these factors. So there's an estimated 2,000 of these words, of these proteins that are involved in cellular communication in our bodies. So we measure only a tiny fraction of them. And we may measure some of the very important ones, because currently we measure the ones that biologists have studied for the longest time. But as we get to more rare ones, it becomes harder to develop assays, but that's what we're working on in the lab. So by and large, if you look at these changes that take place with age, growth and survival factors, and you can call them good factors, they decrease as we get older. While factors involved with inflammatory reactions, with injury, they tend to increase. Now, this is a bit oversimplified, and I told you before, there is no factor that is per se bad or good, but they can assume functions that will be detrimental for the organism. So as we get older, we have less of these good factors and more of the bad factors. But this is all correlation. What I showed you so far is just looking at something that changes and then looking at something that correlates with it. It does not have any causality, right? We don't know whether they're linked to each other. You might as well have the size of their shoes, and that might correlate as well. So the big question is really, are these factors modulating age? Can they affect the age of an organism? Can we take these factors from a young organism and put them in an old organism and change the relative age of that organism. And that's when we come back to this model parabiosis. So I mentioned to you, we surgically connect a young and an old mouse together. And in this case, the old mouse is 18 months of age, which is an equivalent to about 65 years in humans. And we pair them with a 3-month-old mouse, which is equivalent to a 20-year-old. And we leave them together for 5 weeks. So after about 3 to 5 days, the blood starts to mix. And if you had an organism here, a mouse, that makes green cells, we can genetically do that. After a week, half the blood cells in this mouse here will be green, and half will be wild type, we call them. We never see green cells going into the brain. So that's why we argued it's probably not blood cells that enter the brain that are responsible for whatever effects we see. Such cells may be important for other tissue rejuvenation or aging effects that people have studied, but we think cells are not entering, we don't see cells entering the brain, and so we strongly believe that it's mostly soluble factors that we talked about before that are present in the liquid part of the blood called plasma. So here we can now study how is the aging brain affected by exposure to this young blood, to this young plasma, over a period of 4 to 5 weeks. And so what we found is really quite remarkable, and we repeated this many times in blinded studies by different people. And I mentioned to you, Saul now has his own lab, and he completely reproduced these cells with his students now, these effects with his students. So we call this rejuvenation, and you might think that's too much of a word. You can call it revitalization or regeneration. I don't really care. But what we see in the brains of these old mice is that they make more nerve cells again. They make more neurons. So there's higher stem cell activity in the brains of these mice. There's higher synaptic activity. So the communication between the nerve cells in the brains of these mice is higher. There's more chatter going on. And genes that we know are involved in memory formation, in encoding new memories, are expressed at higher levels. So we know a number of genes that are necessary to encode a memory, and these are expressed at higher levels. And then also very importantly, many of our tissues, as I mentioned to you, the blood shows increase in inflammatory factors, and many tissues become inflamed as you get older. Joint pain, for example, arthritis, things like that, is all inflammation in different tissues. And the brain becomes inflamed as well. There's more and more of this low-grade chronic inflammation, and we can reduce that in these old mice simply by exposure to young blood for a few weeks. But we could not test memory in this pair because the memory tests are not really amenable to 2 mice because they have to sort of make decisions, as I'll show you in a moment, and they couldn't really do that. We didn't try it. But because we don't see cells entering the brain, we argued that it's going to be soluble factors. And we decided to harvest blood from young and old mice, spin down the cells, and produce plasma. And this is plasma similar to what the blood center here at Stanford collects and supplies to the hospitals for if you have an injury and you need blood plasma, that's what you would get. So we can produce the same from mice. So young plasma, old plasma. We injected that into old mice. So these old mice got either young plasma or old plasma 3 times, every 3 days for 3 weeks. So they got a total of 8 to 10 injections usually. And then we tested their behavior. And what we found that they had indeed improved memory formation in a number of different tests. So they became smarter again. Mice have the same problem as humans do. As they get older, they get forgetful. And I'll show you a test how we do that. And actually, we're coming to this right now. So the next step was really to ask the question, is there a similar basis of factors in human blood, in human plasma. Can we find similar effects if we compared human plasma from young and old people? Now, we can't directly inject human blood or human plasma into mice because of the species barrier. Also, most of these factors, or a large number of these factors, are conserved through evolution because they're such important carriers of information. Nature didn't change them anymore, and so they're highly conserved. So we can indeed get effects with human factors on mouse tissue. But the immune system of the mouse will reject the human blood if we inject it repeatedly, or the plasma. So we had to resort to a model that is specifically designed, it's genetically engineered, that it can accept human tissues. So these are used, for example, to study transplantation transplantation of human tissues. You can graft in part of a human tissue or human stem cells into these mice. So we could use these mice to test now whether old mice receiving young plasma do better than old mice receiving saline as a control, or young plasma, or old plasma, same age plasma. And the test we use is not unlike this test here. You may all have been exposed to this test, I'm sure. So you want to go shopping, you drive to the Stanford shopping mall, wonderful mall. You park your car in a big parking lot, and then you walk out of your car, you go shopping for a few hours, and then you come back. Where's my car? Now, some of us have this car alarm, so that's a bit cheating. But really what your brain does, it makes a spatial map, and it's absolutely remarkable. Certainly when you're young, you never think about this, and you can't understand why your grandpa, or once you get older, why they can't remember where the car is. Your brain makes a spatial map. It uses cues such as a lamppost, for example, a storefront. There is a Starbucks there, there's a lamp here, over there is the street. And you could just walk out of this shopping mall and your brain tells you it's there and it's going to be there. But as you get older, that encoding does not work as well. And you start to forget, you can't remember. So you have to really remember, when I park my car now, I have to really, okay, I put it here. So that I have to go to the airport later today, and I park at the international garage there, and there's several floors. If I don't remember on which floor I parked, I have to walk up and down the stairs, and then was it here or here, on this side, on that side? So you really have to concentrate to make that map. But anyway, the mice we can do a similar test with the mice. It's not exactly the same, but quite similar. So this is a big table about 3 feet diameter, and it has a lot of holes in it. And first of all, the mice don't like open spaces. They don't like bright lights. So we shine a bright light on this, and this table is above the floor and has all these holes. So the mouse wants to escape from it, and it looks into these holes and finds, oh, I will fall down to the floor. But there's one hole where we put a little tube underneath, and the mouse can escape into this dark place, and it likes that. So we can train it over a few days. It's actually 4 trials per day over 4 days to learn first of all, what is the task, what am I supposed to do, and then where is that escape hole. And it uses spatial cues. So here's a checkerboard, here's a triangle. So the mouse can see these spatial cues and then remember where that hole is. So I will show you an old mouse that was treated with saline. So it's just like any old mouse that, it got placebo. And we put it on this table, and what you can see is the mouse is pretty much clueless. We can watch this. And it just peeks into every hole. That's cheating, right? It just can't remember. And even if we look at at 20 of these mice and we take the average, they have almost no learning capacity in this test. This is a very challenging test. Actually, the original of this, it's called a Barnes maze, had only one ring of holes, but we made it on purpose, we made it more difficult. So now what you're going to see is a sibling of this mouse, so same age, but it was treated with for 3 weeks with plasma from young humans. So it got small injections. It's actually only 5% of the total volume that we inject per injection. In the mouse, that's 100 microliter or 150 microliter sometimes. This would be an equivalent to a unit, 200 mils of plasma in a human, about 5%. So this mouse got young plasma, and if we look now how this performs, it's almost as it first figures out, where am I here, what's going on, and then it walks straight to this hole and escapes. So this is a perfectly performing mouse. I mean, there's some mice that are even faster and go just straight. Now, this is, of course, if you look at the average, not every mouse will do equally well, but overall, there's highly significant effects of this young plasma, suggesting there is something in young human plasma, not just in young mouse plasma, but also in human plasma that can rejuvenate the brain and make this brain function better. And again, we see reduced inflammation. We have all the other benefits that we mentioned. And here is actually the statistics of this, and you can see on this 4th day, there's highly significant effects of doing better than the control mice. And we can also use a different test. This is a test where the mouse gets a slight foot shock in an environment, and it has to remember when it comes back the next day. If it remembers where it was, it will again show this, what we call a freezing response. It will be scared, and we can quantify that. And so again, the mice that are treated with cord plasma, they freeze more because they remember better where they were shocked the day before. So very significant effects of young plasma on memory formation and also on just general factors or genes that are involved in memory formation. So what are the factors that are responsible for these effects? I'm not going to go into detail here, but basically what we're trying to do is we look at studies of normal aging in mice and in humans. We use this parabiosis model to look at the mice before and after the exposure to young or old blood. And then we can make big lists of factors. We measure actually many more proteins than I showed you earlier. We measure now around 500 of these proteins. And then we can try to find the top factors that are associated either with aging or with rejuvenation, with making the animals younger. We can do some cellular, molecular tests, and then we test the individual factors in mice. And actually now we go back to normal wild-type mice, not this special immunodeficient model that I showed you earlier, because here we can now, once we know what the factor is, we can use the mouse factor again and inject that mouse factor and show that it has effects. And we have a number of factors now that we discovered through this pathway that are indeed, if we inject them systemically, so we can inject them in the belly of the mice and they become smarter again and they show these benefits that we see with plasma. So really, the big question is, of course, does this apply to humans? I showed you that mice can be rejuvenated. The tissues can be regenerated, many different tissues, but for us, most excitingly, the brain. It seems that there are factors in human blood, in plasma, in the soluble fraction of the blood, that have similar effects. But can we take plasma and treat people? And We've actually started a company to try to test this idea. We have a trial running here at Stanford that is sponsored, and where we give people with mild to moderate Alzheimer's disease once a week, they get plasma from young Stanford students. I don't know, but the blood comes from the Stanford Blood Center. It could also be from smart high school students. So the big question is, can that young plasma have an effect? And we don't know yet. These clinical trials take a long time. It's double-blinded. I can actually not be involved in it due to conflict of interest, but the trial is running, and at some point we will have results. It's only a safety study at this point, but we hope to see maybe some effects. We do testing in all types of memory tests that are done at the Alzheimer's Disease Research Center that was just funded by the NIH earlier this year. So we have now at Stanford, for the first time, a nationally funded Alzheimer's Disease Research Center. So these patients are seen there, and they get this treatment. We do some imaging. We do some functional MRI imaging to see is their brain working better. And then we also measure some of the factors in their blood to see if there is maybe a rejuvenating response. This will take a long time. The ultimate goal is, as I just showed you in the previous slide, to find individual factors. But for an individual factor to become a therapy, that takes at least 10 years. The plasma, because it's given to hundreds of thousands of people across the globe every day in the ER or in the OR, it's a safe treatment, and we can test it right away. So we're trying to design smart trials, smart studies to see if it is beneficial in people. So what does that all mean? I started with the fountain of youth. So maybe there is something to this vampire story. No, not the vampires. But maybe there's something to this fountain of youth, but it's actually within us. It's the young blood. And somehow, as we get older, this fountain of youth sort of gets down to a trickle. And if we can just turn it on back a little bit, maybe that can help our bodies to stay healthy younger. We don't want to make people live longer. We want people to be healthier as they reach their lifespan. So you want healthy aging with these factors and with this rejuvenation approach. And so maybe that fountain of youth, we can find the factors, or we can even use plasma and turn it on back a little bit and have beneficial effects. I want to acknowledge, so just to summarize, we find that the old mouse and its brain in particular are malleable. So I think that's a very important observation, because it suggests that an aging organ is not set in stone. It's actually still susceptible to changes. Now, I'm not saying we can make the old brain like a young one again. That's not possible, I think, because there will be just mechanical changes that are not reversible. But we can change it back to a younger state significantly so that it has functional benefits. Young blood factors can reverse aspects of brain aging, at least in mice. And old blood factors, I didn't show you that, they actually accelerate the aging process in the young mouse. In this pair, the young mouse suffers from this exposure, consistent with the idea there's more bad factors in the old organism. And then humans have similar factors. And so I want to acknowledge people in the lab who contributed to this work, former lab members, I mentioned Saul already, and several other ones. And then Tom Rando, really, together with him, because he had studied this in the muscle already. My lab was next to his, and so we started talking and tested this idea in the brain. Very important. And several other people funding from the NIH and also at the VA. I have an appointment there. And you can visit our lab website, or you can just send me an email if you have more questions. Thank you very much.
For more, please visit us at stanford.edu.